Oh my goodness. I recognize old friends. This is wonderful. Hi. Oh, Amy, my goodness. Kelly. Oh, Judy, this is great. Francois, Helen. Oh, this is wonderful. Elena. Wow. Oh, we should
So
just,
I guess we'll go ahead and get started. This is really great,
John.
everyone pouring in.
Oh my goodness.
Okay, so hello everyone. I'm Jane Hubbard. I'm the Vice President of the Genetic Society of America, and I'm very proud to welcome you all today for the GSA Award Seminar from Chao-ting Wu, Professor of Genetics at Harvard Medical School, and this year's recipient of the George W. Beadle Award for outstanding contributions to the community of genetics researchers. The Beadle Award is reserved for those whose contributions extend beyond an exemplary research career. Ting's research career is more than exemplary. She and her colleagues have made fascinating discoveries about the relationship between how the genome is organized in 3D and genome function, inheritance, and stability. They've also developed a variety of important technologies, most recently enabling high throughput, super resolution, and genome-wide views of chromosomes. On top of her outstanding research, she's director and co-founder of PG-ED, the Personal Genetics Education Project, which is housed in her lab. The PG-ED team includes scientists, social scientists, educators, and community organizers. They interact with students, policymakers, teachers, filmmakers, communities of faith, and other groups to discuss the benefits and implications that genetics and related technologies have on individuals and on society. The program seeks to promote curiosity and also, importantly, to provide space for open and respectful discussion of the often ethically charged issues that surround genetics and genetics technologies. This approach is sorely needed in our increasingly divided world. I'm honored to welcome someone who's made such a deep impact on both science and public engagement with science. And I'm so glad that GSA has now partnered with PG-ED to develop inclusive public engagement training for geneticists. Ting was presented with her award medal at last month's annual Drosophila Research Conference. And today we have the pleasure of hearing her speak in more detail about her work. Before we begin, however, I have two quick housekeeping items. The first is that all GSA online events are covered by a code of conduct, and the link for that is now available in the chat. The second is to ask you to put your questions for Ting in the chat or use the raise hand function if you wish to ask your question, and we'll get to as many as possible in the Q&A session at the end. So welcome, Ting, and now I'll turn it over to you.
Thank you so much. I'm seeing so many friends here that it's hard to get my head into the feeling of giving a presentation. I want to take screenshots of everybody. It's so wonderful to see you. Thank you for such kind comments. Really, I feel I should take those comments and just transfer them straight to the people in my lab or the members of PGED, because they're the ones who really made this happen. And I will thank you on their behalf, I guess. Oh, but personally, I want to thank you for seeing all my friends here. I'm going to get a little emotional. Wow, I'm getting emotional. Okay, I better move on to science right away. It is so wonderful. This is the best part, I guess, seeing all of you. Okay, so let me open up my talk. This is really such an honor. Whoops. There we go. It really is such an honor to be here, and it was a huge surprise. All right, so we have a number of topics to cover, and I think that I will start with this mystery that arose just about 20 years ago when Gil Bejarano and David Hausler asked whether there are sequences greater than 200 base pairs and that are perfectly conserved between distantly related mammals and human, mouse, and rat. And knowing fixation rates and mutation rates, the expectation was there would not be a single sequence that would be perfectly conserved, but they found 481. And then 2 other papers came out very shortly thereafter confirming that these sequences really do exist. They are 300 to 500 million years old. They're scattered across the human genome, and they encompass a variety of functions, protein encoded functions, regulatory functions. But the thing is that no single one of those functions or all of those functions combined together can explain the ultraconservation. So it is a true mystery to this day. It's considered one of the longest standing mysteries since the genome era. Now, what my lab did was to put function aside and just ask what mechanism would drive ultraconservation, forgetting whether it's useful for the organism. And my lab studies pairing. So we thought, wouldn't it be interesting if ultraconserved elements paired up, they're largely unique, and once a generation assessed each other. And if they found that something was wrong, that would lead to a red flag followed by loss of fitness. In fact, that model would drive ultraconservation. So we've been exploring this possibility. It's a model. We could be totally wrong, but we're exploring it. And I'm going to tell you about some work that we've done. By we, I mean Ruth McColl, who followed on work done by Adnan Durti and Charleston Chang. She worked with Chamath Fonseca, Ren Saylor, and Jelena Ertzig. Sneha spent some time working on UCEs, and right now Tay is our lead UCE researcher. So what Ruth did was to test a prediction that ultraconserved elements do not tolerate deletion or duplication very well. And she took advantage of copy number variants, which are quite young, and asked how much overlap is there between UCEs and Cnvs. And she noted that down, and then she compared that overlap to 1,000 throws of random sequences matched to UCEs in length and number. And then she compared the overlap. Now the black bars here are the distribution of overlaps for her random throws, but the blue bar here tells you that real life Cnvs and real life UCEs do not like each other very much. We were surprised by this because UCEs have been conserved for millions of years, and Cnvs, some of them are only a few years old. So Ruth pursued this and asked, how quickly can UCEs work? And to do that, she looked at de novo UCEs spontaneously appearing in the soma and even in tissue culture. And to her surprise, we saw extreme depletion of Cnvs in all 3 cases. Now, what does this mean? It means that if you took a reference genome and you piled on all the Cnvs of hundreds, if not thousands of individuals who are healthy, there would be gaps. And within those gaps is where you would find the UCEs. This is what the model predicts. But it also says that if you disturb those gaps in a way that disturbs the UCEs, then you would be prone to disease. And in fact, when Ruth explored this with Cnvs associated with cancer, she definitely did not see any depletion, avoidance of UCEs and Cnvs. She's also looked at neurodevelopmental disorder related Cnvs, in particular Cnvs associated with autism. And one study we found particularly interesting. It's a study of data collected by Vulto van Silfoot et al. It represents 118 trios, and that allowed us to separate the Cnvs in the affected individual into those that were inherited from unaffected parents and those that were de novo in the affected individual. And what we found is inherited Cnvs from healthy parents showed a depletion among the UCEs, while those that were de novo in the affected individual were actually far from depleted of UCEs. All right, so what does this mean? We think this means that UCEs may be sentinels, guardians of regions of the genome, and they do this by pairing up once a generation, and if they see a problem with a UCE, then they give rise to loss of fitness. Now, it's a model, and we like to speculate how far it might go. We see no reason why it would not start acting at the moment of fertilization to compare the compatibility of maternal and paternal genomes. And maybe it protects us through our whole lifetime, ridding our bodies of cells that have damaged genomes. Now, as was mentioned in that lovely introduction, for which I want to thank you, Jane, my lab houses the Personal Genetics Education Project. And as she said, the goal of that project is to try and ensure as best we can that all communities, regardless of socioeconomic status, racial, ethnic, religious, or educational background have equal access to information about genetic technologies and equal opportunity to convey their opinions about what is happening. And we'll talk about this later, part of what we do is we interact with producers and writers of television and film to help them incorporate genetics into their storylines. So as we were studying UCEs and thinking about UCEs, along came Carolina Taylor and Jacob Okada, producers and writers, and they had a storyline about a couple that was having trouble conceiving and or having children. And they were looking for a genetic explanation. So we ran the UCE model by them and they liked it so much it became a major part, well, it is the basis of their storyline. So I want to show this little trailer to you. And later on, we'll talk about this film a little bit more. So we like the way they presented the genetics. And later on, we'll talk about the ethics that they raise. So let's see if this works. Okay, open this up. I think I have to.
My grad student accidentally ran your samples along with the UCEs of Neanderthals and Homo sapiens sapiens. Remember, these blue dots, they're yours. They're identical with Homo sapiens sapiens and Neanderthals. And this dot, that's you.
So this is Montel, he's the scientist, and this is the young man trying to have children.
Honestly, it's probably nothing.
Is this why we can't get pregnant?
Let's see what this second sample tells us before we jump to conclusions. Private, totally confidential.
All right, confidential.
The Human Genome Project is a way of exploring our molecular cells. DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains.
I love this scene where he gets excited.
This paper of yours, just about that hypothesis.
Morning to you too.
Yeah, Which hypothesis? Embryo fitness. Hypothetically, compatible UCE profiles lead to loss of fitness in an embryo.
Right.
Hypothetically, could incompatible UCE profiles lead to loss of fitness in an embryo?
If the mothers and fathers' UCEs were similar enough to allow for fertilization and some cell division, eventually embryogenesis can proceed.
So with this loss of fitness, could you still get a positive early pregnancy test?
Hypothetically, sure. If loss of fitness shows up several weeks after fertilization.
Thank you, Rosalind. You've been a great help.
I'd love to meet some of your hypothetical subjects.
Yeah, me too. What's loss of fitness?
So, okay, am I back on? Yes. So this is the kind of things that PG Ed does. We work with producers and writers, and then we try and help them sometimes get their labs to look the way they should look. All right, so now I'm going to return back to the science, and we'll come back to this later. Pairing. So pairing itself, turns out, is a 100-year-old mystery. It's over 100 years old and was started by Nettie Stevens when she discovered in Drosophila that homologs are paired in all somatic cells throughout development. And now here is a picture of Bill Gelbart, who I captured on camera when I was his graduate student, and he was explaining, teaching a class on pairing sensitive gene activity, and was talking about the first 3 genes for which this was shown. The first paper being by Ed Lewis, where he discovered intragenic complementation in a pairing dependent way by thorax and came up with the term transvection. Now, Bill is his paper came out in Decapentaplegic a few years later, showing transvection there. And he also contributed to a paper by Joe Jack and Burke Judge showing transvection at Zest White. I owe so much to Bill for introducing me to genetics and to pairing, which my lab still studies and loves every day. I also want to thank Pam Geyer, who got me through the thick and thins of junior faculty years. And she's just one of my dearest friends. And also I want to thank her for working with Victor Corses and Mel Green to put together this milestone paper on transvection at yellow, where she proposed that enhancers act in trans. So shortly after this paper was published, my graduate student, Jim Morse, who's now at Brandeis, teamed up with Pam Geyer and her postdoc, Jilong Chen, to genetically study transvection at yellow. And indeed, they were able to demonstrate for sure that enhancers act in trans at yellow. And surprisingly, we found that pairing can cause gene enhancers to bypass insulators in cis, because we think the pairing can force an insulator into a loop, an unpaired loop. Now, today we know that pairing has been implicated in a wide variety of phenomena, many of them occurring in mammals. And in all cases, we believe there is some information being passed from one homolog to the other. In my laboratory, several people studied the mechanism of enhancer action in trans, and another slew of people studied the way pairing might happen and looked for the genetic network that supports pairing. There's so many wonderful stories that come to mind when I look at their names, but I need to move on. I'll tell you about Eric Joyce, who worked with Ben Williams to develop a very high throughput fish screening method, which they applied to Drosophila, the whole Drosophila genome, and discovered 40 genes that they think are candidates for promoting pairing. But they found 65 genes that are candidates for anti-pairing. And this finding was anticipated by Tom Hartle when he was working with Gio Bosco, where they discovered that CAPH2 is a very powerful anti-pair. So that, plus Eric and Ben's work, plus Jack Bateman's work with Erica Larschan, who is now at Brown, proposed this kind of model, that the state of pairing is actually a balance between pairing and unpairing forces. And in Drosophila, pairing wins, while in other organisms, unpairing wins. So when do those genes act? Well, here I want to talk about this beautiful study that Jack Bateman did using compound chromosomes, which allowed him to make embryos that lacked entire chromosome arms, one at a time. And because those embryos could initiate pairing perfectly well, he was able to rule out in 5 simple crosses, or maybe it was 6, that the initiation of pairing in the embryo does not require any zygotic gene activity. We proudly published this in Genetics, which is one of my favorite journals. We published our transvection papers there. We just sent them straight to Genetics. Now, when Jack left the lab to start his position at Bowdoin College, he left a huge emotional hole in my group. And I discovered one day that people were planning a sleepover at Jack's. So we had this sleepover, and that turned into an annual affair where pairing fans get together. This, we have a variety of names, and we use this last one when we put it on our formal CVs, because the other 2 just don't have the same weight. So here's a picture of our first sleepover with Jack's entire family there. And then our meeting in 2013 was held by Gio Bosco at Dartmouth. This is a card we put together for Pamela Lewis to let her know that pairing was fine and doing well. This is a meeting at Harvard where we, the entire group, learned how to juggle at the same time. And this is a portrait of the meeting that Jack Bateman organized at Bowdoin. And I don't have pictures for our meeting at Laurentian in 2018. And then just before the pandemic, so Thomas Merritt organized that one, and Michelle Longworth organized our meeting at Cleveland Clinic. So we can't wait for the pandemic to be over and for all of us to get back together again. But what does pairing look like? So here I want to tell you about work done by Yelena Ertzeg, who is now at UConn in her own faculty position, and Jumana Haj-Abed, who is still in my lab, Anton Golo-Barocco, who now has his own lab at the IMBA in Vienna. He was working with Leonid Murney at the time, and Brian Lajoie, and was working with Job Decker. So what they did was to make hybrid Drosophila so that we could SNP resolve the maternal-paternal chromosomes. And then here's a Hi-C map that maps, it's a heat map of the interaction frequencies between chromosomal regions. And like all Hi-C maps, it has its major diagonal. But because we could map the maternal-paternal chromosomes separately, we get an off-diagonal diagonal. Now, as we went along the genome and measured the amount of pairing, which is what this off-diagonal stands for, we discovered that the degree of pairing could be defined, the distribution of pairing could be defined by two curves, identifying what we think is a loose pairing structure and a tight pairing structure. How do they look on the Hi-C level? So here's that off-diagonal diagonal, a close-up. For tight pairing, we get a really strong diagonal. For loose pairing, we lose that diagonal entirely. And this had been seen only once before by Egan, when he was looking at polyT nuclei where sister chromatids pair very well together. What does this mean? Well, we think tight pairing might indicate railroad track pairing, where the homologs really are aligned quite well. And loose pairing, we think, is kind of a laissez-faire, let's just get close together, but not that close together type situation. Well, what Yelena and Jumana then did is to see how these types of pairing correlated with the A and B active and less active compartments as defined by Hi-C, and how they measured up relative to highly transcribed regions and lowly transcribed regions. And the first thing they noticed is that highly transcribed regions are associated with tight pairing. This really went very nicely with some modeling done by Lee et al. And Frank Albert's group, by Jordan Rowley and Victor Cortez's group, and Kyla Vietz in Bob Johnston's group. So that was exciting. You know, it's nice to have data coincide. But what's interesting is that tight pairing actually correlates pretty well with lowly transcribed genes too. So what we think is that maybe pairing can poise a region to either be silenced or turned on. It's a form of genome-wide poising of chromosomal regions for regulated activity. In contrast, loose pairing is almost exclusively associated with very lowly expressed genes. All right, now what does this mean in terms of structure? So here's where I want to move on to imaging. Our imaging technology was really made viable by Brian Beliveau when he was a graduate student. He now has his own lab at University of Washington, working with Eric Joyce, who is UPenn, I should have said that before, and Ben Williams, who is at Helms Charitable Trust. What oligo paints are, the technology we use, are computationally designed oligos that have a region of genomic homology and two regions without genomic homology, which we call main street and back street, and to which we can park all kinds of functions. And what Brian did was to take advantage of that structure and implement super resolution imaging. In one case, OligoStorm, he collaborated very closely with Alistair Bediger, who's now at Stanford, when he was in Xiaowei Zhuang's lab. And for oligo-DNA paint, he collaborated with Meyer Avendano and Ralph Youngman in Peng Yin's group. Ralph actually invented DNA paint well before oligo-DNA paint came along, and he is now at the Max Planck. And then Stephen Wang and Xiaowei Zhuang's group demonstrated the ability of oligo paints to be used for sequential fish. So I'll describe for you OligoStorm, which is a super resolution strategy we use most frequently in the lab. What OligoStorm does is it picks up single blinks that are temporarily and spatially separated and records those blinks in a gigantic tables of XYZ coordinates, which then we computationally put into a three-dimensional structure where each one of these spheres represents a single blink from a single fluorophore. What you're looking at now is a loop that's 290 kb in size with two 10 kb anchors. I won't tell you how variable this loop can be, that's another story. This technology can also be, oops, I should tell you this was work spearheaded by Guy Neer. He now has his own lab at UTMB and in close collaboration with René Farabella from Marc Martí-Granome's group, Cynthia Perez Estrada, who did the high C analysis we needed. She is in Erez Lieberman Aiden's group, and we work very closely with Brucker, where in this case, we worked closely with Carl Ebeling and Jeff Stuckey. And I want to mention Dean Lee, whose work I won't be talking about today, but he is an expert in distinguishing maternal from paternal homologs using SNPs. Now, this technology can also be used to walk along megabases, tens of megabases of the genome. So here is a 8.16 megabase walk along chromosome 19 in a human cell. And because the Brucker microscope gives us the depth we need to get through a nucleus, we most often can catch both homologs. So we can compare the maternal-paternal homologs. And right now, I won't talk about it very much, but there's a great effort in the lab going on to try and be able to walk through the entire genome, human genome at super resolution. But now that we have two homologs here, let's get back to pairing. What Jumana has done is to walk along the Drosophila genome through loosely paired regions and tightly paired regions to see if they are structurally different. So right now, I'm showing you 4 regions, 2 that are tightly paired, flanked by 2 that are loosely paired. And she's looking to see, is there a common feature of loosely paired regions and tightly paired regions? And in this next image, I'm showing you these 4 regions. These 2 are tightly paired in the middle. Those on the side are loosely paired. And you can tell right away, regardless of size, that loosely paired regions and tightly paired regions seem to equally entangle their homologs. This was not what we expected. Here, Jumana used a probe to distinguish the paternal homolog from the maternal homolog, and she falsely colored the paternal and maternal pink and blue, respectively. So what does this mean? We think that it means that loosely paired regions are maybe not that different from tightly paired regions in the amount of compaction, but Jumana pressed forward to see if the images could give us some hint as to whether there's a structural difference between these 2 types of pairing. And I think she has found such a difference. So here is that sort of half the transcription map that I showed you before. What Jumana does, I loved her strategy. She labels each of the quadrants, and I'm putting those labels to the right so you can see what happens between how tightly paired regions and loosely paired regions differ when we graph those regions by their radius of gyration normalized to genome size. I think you'll see pretty clearly that tightly paired regions have a larger radius of gyration than loosely paired regions. Now, one interpretation of this is that tightly paired regions are actually not as compactly packaged as loosely paired regions. And one possibility of this is that tightly paired regions are just not as flexible. They're a little stiffer. We're excited about this because in fact, there's evidence that this might be true from Paul et al.'s study looking at sex-sorted Drosophila embryos. They believe that the single X in males is more flexible, and that might be related to its higher activity to paired homologs in paired X chromosomes in the female embryo. This idea that pairing of the X chromosomes or unpairing of it can account for dosage compensation is it's an age-old teaser in our field, starting with a paper from Tom Klein's group that hinted at this possibility, or at least told us we should think about it. And most recently, a paper just came out, Galuzzi and Prudhomme, just a few weeks ago, that has reproposed this model. So I can't wait to see where this goes. I seem to be drawing on my screen. Okay. And actually now I'm going to leave the science and return to PG Ed. There's a lot to say, but this next piece I really want to spend time on. So this movie, we were excited about this movie because what Carolina and Jacob did is they passed on genetic information to their audience, or you can get this from Amazon Prime. And in addition, they raised some really interesting ethical issues about incorporating technology, genetic technology, into people's lives. I feel that this movie makes us ask who we are, makes the audience ask who we are. And it really addresses, makes people ask, what does it mean to be genetically different from your parents, from your siblings, from your neighbors, from your distant neighbors? And it makes you ask, does it matter? Well, in this case, it mattered to this couple because they couldn't have children. And then at least for me, it made me think about how much responsibility do I have as a geneticist to helping guide genetic technologies into the knowledge box of non-geneticists. How much is that my responsibility? And so I like this movie because it has something for everybody to think about. Now, it's because of questions like that and watching what has gone on over the past year in our country and in the world that drove us to actually approach GSA for a partnership, because GSA has this reach and is one of the oldest genetic societies that we have in the world. And I just want to tell GSA, it's been such an honor to work with you. And we are overwhelmed by the force of your enthusiasm. It's just, I think we're very excited about what can happen from this. And for those in the audience who are interested, please do feel free to go to our website and see what the next events are coming up. There's no commitment. You can just join to see what it's like. And help us keep the conversation going. But in the meantime, I can tell you very briefly now what PGED does. This is just a sampling. So we go to schools, we meet with students, we do teacher training. Sometimes teachers come to us. But in addition, we put online lesson plans that cover topics like this. And these are lesson plans that are ready to go. Basically, a teacher doesn't have to do any research. They can just look at what's there and be ready to teach. We don't, however, teach GATC, Punnett squares, recessive dominance. Our goal is basically to get a conversation going. And the way we do that is to provide information. And we're very careful not to promote genetics. Our goal is to help people feel comfortable discussing genetics. And we learn as much as we give. I think we learn more than what we give. The conversations that we have are so enriching of us. And here are some pictures of the students that we've worked with. Students like to be revisited. So for some schools, we go back and revisit them. This is a game that has dropped a little bit. I'm hoping it comes back. If you answer 5 questions, maped.org with a dash, then you can, everyone always gets 100%. Then you get to map yourself on a world map. Now, Adam Steltzner, who was the chief engineer that put Curiosity on Mars and recently just put Perseverance on Mars, he played this game and he mapped himself on one Bradbury place on Mars. So we like to tell people we're probably one of the first or the first interplanetary educational program. And as you know, we work with film and television. In addition to the Anya film, we had a role working with the really incredible script writers for Grey's Anatomy. And we had a little bit of a role in helping Bailey get her genome sequencing facility off the ground. She didn't have to write too many grants though. They said, you know, does it take a year or two to build a sequencing facility? And I said, oh my gosh, it takes years to get funding. So in the script, she inherited a tremendous amount of money. And next thing you know, there's a genome sequencing center. This organization I work with, the Science and Entertainment Exchange, has writers' retreats. You spend 2 or 3 days with writers, 50% scientists, 50% writers, and you work on scripts together. What else do we do? We hold congressional briefings. This one is discussing the use of genetic technologies to protect astronaut health in space. These briefings, we're told, are successful if you get 26 people. Our briefings are usually filled to standing room and they're bipartisan. So we're hoping to get that going. There's so many things that can be done in the area of genetics, and we work with communities of faith. I will say the enthusiasm of communities of faith, even when they don't like the technologies we talk about, is impressive because they believe that especially to take care of the disenfranchised communities. So that's where we align so well with communities of faith. And I think I am at about 30 minutes. So I will give the many, many acknowledgments. There should be so many more people on these slides. It was very hard. This is the hardest part of putting this presentation together. But it's easy to thank individuals. So I really want to thank the current staff, Marnie, Robin, Mohammed, Nadine, and Dana. They are incredible, great sense of humor, and tireless. We have many alumni at PGED. I want to thank our alumni staff and the interns that have worked with us, and very big thanks to Jack Bateman and Dana Waring, who are co-founders of PGED. PGED gets funding through NIH and through our collaborators, and I want to, though, single out Cliff Tabin, who's the chair of our Department of Genetics. He has pulled us out of the hole many times. He is an extraordinary chair. He told us that he didn't feel he could run the Department of Genetics without something like PGED, and we owe our existence to him. PGED gets funding through in-kind support, grants, gifts, corporate donations. I really want to thank some individual donations from Jennifer Dowden and Jamie Kate, and from Lei Lee. And for disclosure, I will say that Ashley George, my husband George Church, and I also support PGED. And then right up front and center, thank you, GSA. We are so excited. It's one of the brightest things that happened in this very past dark year. For research, I think I mentioned the researchers whose work I described. So these are the people who I didn't mention: Hoy, Shamtanu, Antonio, Sarah, Irene, Dean, Nuno, Mohammed, Faye, and Connor. They are part of the next front of researchers and imagers who are trying to make our imaging go genome-wide and go into tissues. Irene is leading the charge on integrative modeling and teaching us while she's at it. So many thanks to this group. I think I've mentioned all these others, but in green over here, I really want to thank Sunny, Nick, Faiza, Yiming, Caroline, and Hian also for supporting work in the past on imaging. I have so many collaborators. I'll have you read their names. I'll just mention the PI's names. We worked with the Talkowski, McCarroll, Murney, Decker, Wren, and Zhang Labs on UCE and pairing. My close collaborators ongoing right now are Mark Mati-Renom and his group, Erez Lieberman Aiden, Nicola Naretti, George Church, and Peng Yin's groups. We are hoping to accelerate imaging and make it much more accessible to our colleagues. I'd like to thank Peng and Xiaowei for early work getting our super resolution imaging going, Jeannie Lee, William Shi, Jose Onichuk, and Fred Bantigny for help in making lines and making probes and in computational analysis. And finally, my funding comes from NIH, a variety of sources of NIH. I want to acknowledge Bruker. We're close collaborators. They recently have given us a sponsored research agreement award, so that's been very helpful. We've worked closely with RECOR and with microarray early on. Jean-Marie Rouillard helped us design the first oligopaint, or synthesize the first oligopaint libraries. And then just want to disclose that Acuity Spatial Genomics is a company that just got started this year. I'm associated with it, and our goal is to bring the costs down of imaging, make it more accessible, and hopefully move it into a zone where people can just, I won't say plug and play, but can apply to the research without a lot of effort. And with that, I want to thank GSA. I once again want to thank my advisor, graduate advisor, Bill Gelbart, and Pam Geyer for helping me to get here. So thank you very much.
Thank you very much, Ting. That was fantastic. I see the questions are pouring in. If I see a hand raised as well, why don't we go ahead, Tom Klein, if you'd like to ask your question.
Yeah. Hi, Ting.
Hi. It's so nice, Barbara. It's great to see
Long
you.
time. I learned a lot, especially about PGED. I did have a question. I'm wondering, is there something, I haven't been following this whole area, I apologize. And the question is there something special about polythene chromosomes? That is, do they use a special set of the pro-binding genes? Or is it just a simple consequence of the pairing anyway in Drosophila?
So you're wondering, like, what's keeping them together?
Was there something special about polytonization, I guess is what I'm wondering, or is it just a secondary consequence of the fact that the chromosomes are paired? I
wish I knew, Tom. I'm pretty sure any question you ask, I'm going to say, I wish I knew. It's great to see you. Hopefully, with some of these candidate pairing promoters and pairing anti-promoters, we will be able to begin to dissect that. I know that CAP-H2, this is through work done in Gio Bosco's group, when you apply CAP-H2, the polyteins are not so close.
Do you have any idea why there should be so many of these genes? It's sort of mind-boggling that there'd be a huge cluster of antis, competing with a huge cluster. It just seems crazy.
It is, there's a gene regulatory approach to that. But part of me wonders about this idea that when you pair, you're a little stiffer, you're a little more open, and maybe pairing it both helps in gene regulation, but helps in creating some kind of structure, maybe the meaning for which we don't even know right now. We are getting more and more interested in the structural outcome of pairing, not just its regulatory outcome. Yeah. I'd love to talk to you about this anytime.
Thanks. Likewise.
Oh, it's so good to see you.
Great. So we have a question in the chat from Zhao Yang Yu. She says, he or she, I'm so sorry, says, great talk. Homologous pairing is a huge part of meiotic progression. Just wondering if you've looked into the pairing mechanism in meiosis. Would you anticipate a similar transcription regulated pairing occurring during meiosis?
Wow, I love those questions. I think it would be so interesting if transcription were involved. I don't know the answer to that. And we haven't yet looked at the intersection of meiotic pairing and somatic pairing, but I think that it makes so much sense. The one thing we have found, oddly, people thought, for those of you who aren't Drosophilus, that Drosophila didn't have to worry too much about pairing its homologous chromosomes in meiosis because it's paired, those homologs are paired all the time. Well, to our surprise, when Eric Joyce looked at the meiotic cells and the meiotic germline, that's the one place where homologs are not paired. So the interface is not what we thought it was, it's the opposite. And we're wondering if that is on purpose, and that actually is what links them together. The transition that keeps them, the transition point, maybe what makes them mechanistically linked. We don't know.
Okay, great. So our next question is from Doug Koshland.
Hi, Doug.
How you doing? So I'm curious, when you make your models for using structure to propose mechanism, in the cohesion looping field, one of the big things that came out of that was that these loops are incredibly dynamic. And I've always felt that people who are proposing gene regulation for looping haven't incorporated that, because it's really hard if you think a loop is excluding an enhancer from talking to a promoter. If it's only present 5% of the time, that gives the other 95% of the time that enhancer to talk, that promoter to talk to a lot of other enhancers. So when you generate these structures like tight pairing and loose pairing, your data is, if only 5% of the cells have that particular structure, it's a little bit hard to make a model, about. So how do you think about dynamic and your readout for structure and dynamic structure?
So I'm going to start at the loop and go backwards, and start off by saying Drosophila may differ from all other organisms because its pairing, we think, is actually, it's more stable and it's more extensive. So what I'm going to say now is based on Drosophila. It may not apply to other organisms. But let's first start with dynamicity. When we look at our structures by super resolution, we see incredible dynamicity, so much variability. But because the homologs generally are similar to each other, we don't think it's random. We think that the genome is quite active in its structure. So I entirely agree with this question about loops. So what's happening in Drosophila? Well, there I think the pairing is much more stable. And in fact, because Drosophila doesn't have the same probably CTCF cohesion loop type mechanism, it may not have it. We actually think that the looping is highly dependent on pairing. I took these images out. So imagine you have two homologs paired like this. Now imagine where my band is there's unpairing. And that unpairing spreads a little bit from the middle of my arm to the middle of my hand. If then we have anti-pairing, pull those two apart, the chromosome will come together. And we will have a loop, but it won't be a cohesion mediator, CTCF loop. It'll be a pairing. The ends of the parts that don't unpair will come together. And the enhancer and promoters, if they're at the base of that loop, will be held together by pairing. So I think in Drosophila, maybe it's not so dynamic. But in other places where you don't have pairing all the time, maybe there's memory. Or maybe you just don't have to be so close to your enhancer or promoter all the time, or silencer. What do you think, Doug? I feel like
I think the concept of memory has to be imposed in here somehow, but I'm waiting for somebody to propose a mechanism of what the memory is. So.
Well, we're actually wondering if some of that memory is a structure. Maybe not a structure we would recognize with our eyes straight off, but it could be that this variability and this motion slowly sorts regions into different, if we did cluster analysis, they would go into different groups, and it'll be difficult for, okay, let's look at cluster analysis. You have structures here and structures here. It'll be difficult for a structure here to jump into that structure. It would have to backtrack and come down. And so maybe what we're looking at is a sorting of chromosomal regions into sets of structures that are, however, unable to interchange. But right now, when we image, it looks like just a lot of variability.
Okay, we have a question from Astrid Haas, who asks, does pairing require RNA? Okay, there you are, Astrid. Would you rather ask in person?
I'm sorry, I hope you forgive my naive question, but you were talking about structures, and I'm wondering if it either requires or leads to the formation of RNA-DNA hybrids or structures.
So, boy, I think I'm almost completely, I haven't been able to answer any question except I don't know, and I don't know about this, but I do know in meiosis, I think in fungi, sometimes RNA is very much involved. And in fact, RNA is a very interesting possibility for not only pairing, but sequence matching. And early on in the transvection, I haven't seen this too much lately, but early on in some transvection studies, it was proposed that RNA from one homologue may actually be the connection to the other homologue. So I think those are all still on the table. Also on the table. And if anyone has answers to some of these questions, please speak up. I'm totally curious what you would say too.
Okay, we have another couple of questions from Nikhil Hajirnas. I'll go ahead and read them if, okay, so the first one is, first of all, Nikhil says, wow, amazing talk, wonderful efforts for education. What is your take on factors that cause transgenerational inheritance? You think pairing mechanisms are somehow related to it? It's a very broad and vague question, but I'd love to have your insights. And the second question is, can extra richness of availability of boundary proteins contribute to pairing in Drosophila? And in higher animals, is this function taken over by a secondary function by a transcription factor?
Wow. Okay. So these questions,
they're in the chat, but I can restate if necessary.
I think I found it. My head, these questions have put my head in many places. So transgenerational inheritance. Yeah, we are wondering, okay, I want to make sure I read that question. Okay, so there's so many things that come up with that. So I'll just take one take. If I don't answer your question, you can ask me again. I think there may be many, many ways in which transgenerational inheritance can happen. We're discovering them kind of one by one. Why wouldn't an organism want to pass on information from one generation to another? I see every reason why it would. So maybe we're just at the tip of the iceberg here. One particular thing we think about a lot is meiotic pairing. Pairing we think about as being there for recombination, for segregation, but pairing is a structure. And now we know structures are quite variable. We know structure almost certainly has some effect on the activity of a region. Wouldn't it be interesting if structure is one of the things that's passed on transgenerationally, or even from one homologue to another? Maybe we're talking about more than just transgenerational inheritance. So the factors that we're looking at that have to do with pairing, I would love it. it would be so interesting, it would make sense if some of them also partaked in things like transgenerational inheritance. And if, so we sometimes talk about in the lab how if we, eventually we can image the genome of humans very easily and inexpensively, that if we did the same thing for structures we do for genome sequencing, we may find lineages. So your family lineage may have a different organization than mine suddenly. It may be a heritable feature that goes hand in hand with sequence. And so, okay, I'm going too much on transgenerational inheritance. 2, can extra richness of availability of boundary proteins contribute to pairing in Drosophila? Oh yeah, so it turns out that if you look at tight pairing, it's enriched in architectural proteins and insulator proteins. So yes. And in higher animals, is this function taken over as a secondary function by transcription factor? Great question. I don't think we know the answer, but it is striking. Not only do mammalian cells not pair their homologs, there is evidence somehow that there's some studies, I could go into those, where the homologs are kept further apart from each other than you would expect. So there, maybe you got rid of pairing proteins and you replaced them with some other factors. I don't know if that was your question, Nikhil.
Yeah, so I was also wondering, is it also because of the availability of intrinsically disordered regions in the transcription factors as you go higher and higher? Could that be a reason that it's keeping it in a separated domain and not paired?
Yeah, actually, I think the disordered domains are, there are a lot of, I think that you and I think the same. I think there's a lot to come from those disordered domains. Maybe they look disordered to us, but that they're completely what they need to be.
Right.
Yeah.
Also, I would like to come back to your first insight that, you're saying that pairing could be structurally conserved across transgenerational, but isn't it amazing that in a haploid organism, you have the 2 gametes that now have only haploid set of chromosomes. So how is the clue for pairing retained over there?
Okay, this is going to be kind of a cheating answer, but we think even in haploid organisms, once you go through a round of replication or at the replication fork or the replication bubble, you have 2 sisters and pairing could happen right there. So it may be in haploid organisms, if there's something that really needs pairing, it will happen after replication. That's from someone who's just pairing obsessed.
Okay, we have another question in the chat. Alexander Patberg says, amazing talk. I'm wondering if you see a possible application of your model in mammalian synthetic biology in regard to gene regulation. Is there a way to further upregulate gene expression this way? In other words, I guess by promoting pairing. Correct me if I'm wrong, Alexander, but I'm guessing that's
right.
I think that's a really wonderful idea. Yes. In fact, I think pairing could be used to enhance homologous recombination gene editing. But if we can figure out how tight pairing can turn genes on and off, I think that would be a very interesting thing to explore, for sure. Thank you for bringing that up.
I had a quick question, and then we'll go to Giovanni Bosco's question that just came in. I couldn't help but notice that maybe the slide went by too fast, but I thought I saw ribosomal proteins on your list.
Oh.
And I just couldn't fathom what they would be doing, interfering, or how they would be interfering with the pairing.
So
did I miss that, or were the names Drosophila names and?
On the RP
RPLs and RP.
Yes,
there was
yes.
yeah.
So I don't know. We still have to figure that out. We've only just started to look at some of the candidates, but I have to tell you that we got so many. Maybe if there were 3 or 4, we'd have gone. There's so many, so we haven't really dug down into them. But yes. And why would ribosomal proteins have anything to do with pairing? I don't know. Except
okay,
maybe,
great.
Except that maybe pairing. I imagine that if pairing is a very active process, maybe it's indirect, but I don't know.
Okay, so Giovanni Bosco says, a wonderful talk, Ting, thank you. Do you think bits of polyteins can template non-polytene chromatin to pair with it in vitro? Do you know if anyone has tried that?
Geo, that's such a Geo question. Geo, I don't know. What a great idea. Are you thinking in vitro, to pair with it in vitro? Why not? I guess you could try that. And we could use oligo paints to assay whether it's bound there or not. That's a really interesting idea. Gio, did you want to answer the question a little bit more about polyteins and pairing and unpairing? Because that's your work that I summarized so poorly.
No, you did a great job. Thank you, Ting.
That was very generous of you, Gio.
Okay, well, if there aren't any further questions, I don't know if I'm supposed to hand it over back to GSA at this point. Yes, Jessica is reminding everybody in the chat to please join us on May 5th to hear Edward Smith's talk. He's the recipient of the Elizabeth W. Jones Award for Excellence in Education. The talk will be Culturally Aware Research Education, Pay Attention to the Differences. So I encourage everybody to register for that. And many thanks, Ting, for a really tremendous seminar and presentation. And congratulations once again.
Maybe thank you for this award. Honest goodness, I should split it among my lab and the PG Ed. And I just want to thank you for bringing all my friends here on one screen. It's been wonderful. Thank you all for visiting. This is great. Thank you.
Thank you for joining us today.